Okay. So how are you all? How are you all, students? All are fine? Okay. So let us start the class. So in the last class, we have completed the biotechnological process and the principle, right? So from today, we are going to start the biotechnological applications. Okay. Let us start the class. Okay. 1 minute. Yeah. So before going to start the class, as usual, there is a question for you. The question is, which one of the following is used as a best genetic vector in plants? Okay. The question option is Bacillus thuringiensis. Option B is Agrobacterium tumefaciens. Option C is Pseudomonas putida. And option D, all of the above. Okay. Genetic vector I'm asking. Okay. Answer me in the chat box. And you can answer me in the poll also. I will launch the poll. I need answer from you all. Okay. Okay. So I have got the answer. Okay. So very good. So everyone answered to this question. So yes. So the Agrobacterium tumefaciens, right? Agrobacterium tumefaciens is a natural vector. So that we will use for only in the plants, right? We will not going to use it for the animals, right? So option B is the correct answer. So that too in the only plants, right? Okay. So next we will come to the biotechnological applications. Okay. So our biotechnology has application in all the fields. Okay. See here. First I will tell you, and we are going to study only 1 or 2 applications in the text. Okay. Yeah. So let us study one by one. Okay. Yeah. See here in the biopharmaceuticals. Okay. 1 minute. Okay. So in the biopharmaceuticals, in the therapeutics, and in the diagnostics, genetically modified crops, processed food, and bioremediation, and waste treatment, and the energy production. Okay. So this all these the application of the biotechnology, but we are going to study only few. Okay, not all the things. Don't take worry. So we are going to study only the few here. Okay. Let us study. Okay. So this is the three critical research in the biotechnology. So what are those? First, what we will do in the biotechnology? So we will create an enzyme, right? So whenever we are taking the enzyme, we'll, for example, you will create the curd in your home also, right? In a home also, you will create a curd. That bacteria is consisting of the so many enzymes in it, right? So whenever we create the some other bacteria will also be there in the curd, right? Other than this lactobacillus. So whenever we create in the biological that is in the industry level, if we do that means we have going to identify only the particular bacteria, only that bacteria is going to be used there, right? And if particular enzyme is going to be used means that particular enzyme, purified that enzyme we are going to use in the biological product formation, right? So the first thing is the best catalyst to form improved organism, usually the microbe or the pure enzyme, right? So if this bacteria is going to give this particular product means, if some other bacteria is also there in that means, it will decrease the product formation, right? So that is why in the industrial level or in the biotechnological process, we will purify that microbe or the specific enzyme. Okay. So then second thing is create optimal condition through the genetic engineering for the catalyst to act. Okay. So I have told you that if any enzyme is going to work means it must have a specific pH or the specific temperature. That we will call it as the optimum pH or the optimum temperature. In the last class also we have studied about the bioreactor, right? In the bioreactor, what it is going to be happen? So we are going to give a good condition for the product formation, right? So we are going to give a oxygen supply, and we are going to give a specific pH, we are going to give a temperature that is specifically for that enzyme, right? So then we are going to give a, then we are going to get a good yield, right? So that is the one process. Second process. First one is the pure microbe or the pure enzyme. Second one is the optimal temperature or the condition for the product formation. So third thing is downstream processing technologies for purifying the protein or the organic compound. For example, if you got some product, okay, have you think that in the bioreactor, which are the products we have got that? So that is only the final product? No, right? So it is consisting of the contaminants, right? So we are going to remove that contaminants and we are going to get a pure products, right? We are going to get a pure product. Later the downstream processing, not before only. Okay, later the downstream processing we are going to get. So this is the another thing. Okay, so these all the three things. One is a pure micro, pure enzyme. Second one is a good condition for creating that product. And third thing is for the giving that in the pure product form. Okay, without the contaminants, we are going to get a pure product, right? So these all the things is important in the biotechnology. Okay. Yes. So then we are going to study about the biotechnology application in agriculture. Okay. Student, here I have to tell that. Have you read about the MS Swaminathan, sir? Have you read about MS Swaminathan? Can you tell me who is MS Swaminathan? I need answer from you. Can you give me who is the father of? Okay, I only told the answer. Okay. So MS Swaminathan. Okay. So MS Swaminathan is the father of the Green Revolution. Okay. So we know that. So day by day, the population is going to be increasing, right? So for that, what is the solution? So whenever the population was increasing, so to thrive for the food production is also being increased, right? So what are the things we have to do for the increasing the food production? What are the management practices we have to do for the good food production? See here. First, we'll learn about the MS Swaminathan. Okay, MS Swaminathan. I think it is visible, right? I think it is visible, right? MS Swaminathan. MS Swaminathan is called as the father of Green Revolution. Green Revolution. Okay. Green Revolution. He was increased the agricultural products. Okay. Agricultural yield to the 3 times. Okay. 3 times he was increased the agricultural yield. Okay. So that's why we'll call it as the Green Revolution. Okay. See, let us study the application in the agriculture. What are the application in the agriculture? Okay. So I have told you, right? The population was increasing means the food production has to be increased, right? So there are 3 methods we can use. One is the agrochemical based method. So that is the agrochemical based agriculture, right? I have told you that. What is the agrochemical based method means? In the agriculture, we are going to use the fertilizers and pesticides. Okay. Fertilizers and the pesticides. You know, these are the chemicals, right? These are the chemicals we are going to use this for the increasing the food production, right? For example, if the plant is going to be affected by the pest or any insect, means it is going to give a low yield, right? So whenever we use the good fertilizer and whenever we use the pesticide and the insecticide, it is not going to be affected by any pest or any insect. Then obviously it is going to give a good yield, right? Obviously it is going to give a good yield, right? So this is one thing, but the agrochemical based method is also has some harmful effect. Okay, it is also having some disadvantage, right? So what is this disadvantage? See here, these chemicals are costly, right? So these chemicals are costly. Okay, so what are the disadvantages? First one is these chemicals are costly. These chemicals are costly. Okay. So second thing, these chemicals are costly, right? So farmers, it is very difficult to buy this. Okay. And second one is Obviously, the chemicals means it is going to cause environmental hazards, right? Environmental hazards it is going to be caused. It is poisonous to the environment also, right? So that's why this thing, it will give a good aid, but it is harmful also. Okay. So then second thing came is organic agriculture. So how we are going to do the organic agriculture? So we are going to use the organic manure. Okay. We are going to use organic manure. Okay. Organic manure we are going to use. And so thereby increasing the food production, thereby increasing the food production. For example, you can take a cow dung for as a fertilizer we are going to use. So, but what happened? These all the things by the organic agriculture, it will not give the more yield. Okay. So then also we are require some other solution, right? So finally we have got that solution. What is that solution? So that is the genetically engineered based agriculture. Okay. So what we are going to do here? Okay. So we are going to genetically modify the plant here. Okay. So let us study these all the things. So how it is useful in the agriculture. Okay. See here in the genetic engineering, what we are going to do. So genetically modified organisms, like we are going to do the GMO plant or GMO bacteria or GMO animals. Okay. So one by one we will learn this. Okay. Okay. So let us study about the application of the genetic engineering in the agriculture. So can anyone tell me which are the applications you know? First one, we can tell that pest resistance. We can make the pest resistant and insect resistant plant, right? Insect resistant plants, right? And can you tell me, guys, remember what are the applications are there in the agriculture? We can produce the disease resistant plant also, right? Disease resistant plant, right? And you have to remember here, we can produce a plant which can tolerate the abiotic stress also. Okay. We can create a plant so that is resistant to abiotic stress. Abiotic stress. What do you mean by this abiotic? You can take a temperature, or you can take a water content, or you can take a you can take what we can call as cold, temperature, and the water. So these all the things are the abiotic stress. Whenever the water was less, so we know that some particular plant require the particular amount of the water to grow, right? Otherwise, it will give some disease condition, right? Or if the plant will be grow correctly only in the particular temperature, otherwise it is not going to be grow, right? So we are creating the plant, if anything, so that is in the it can tolerate the abiotic stress. If the less water is there, then also it will grow. If the high temperature is there, then also it will grow. And cold is there, then also it will grow. Okay. So like that, we are going to produce a plant so that will tolerable to this abiotic stress. Okay. So then the another thing. So what we can do? So another thing is the post-harvestal loss. Okay. Post-harvestal loss. So this is very, very important for the farmers. So what they have did? So they have cultivated the crop. So then they have produced the crops. So after that, they have to sell that, right? After that, they have to sell in the market. So they will wait for the good amount to be increased, right? Then only they are going to be sell that vegetables. So that should be not be ripen, or it should not be waste, right? So whenever what happened, if we harvested that vegetables, then we are kept for 1 or 2 days. Then it is going to be spoiled, right? Within that time we have to give to the market, right? So we have created some vegetables or some of the agricultural products, so that can be the post-harvestal loss. Okay, so we are reducing the loss of that. Okay, so for example, you can take this tomato only. Okay, have you heard that whenever we are in childhood, or you can ask your fathers or mothers. So whenever we buy the tomato, so before the this GMO crop was come. Okay, before this tomato was came. So if we take the if you buy the tomato from the market, it is going to be spoiled within within the 3 to 4 days. Okay, okay, within the 3 to 4 days. But now what happened? If we take the tomato, have you observed that if you buy the tomato, it is going to be, we can keep the tomato till 5 to 7 days, okay, without ripening. Without ripening, I'm telling, okay, without ripening, and we can the flavor and texture of the tomato will be same. Okay, it is not going to be ripened. Okay, that is the main reason. So here they have introduced it, or they have the reason for the tomato ripening is one of the enzyme. Okay, that enzyme name is polygalactourase. Okay, polygalactourase enzyme. Okay, so in the biotechnological process, what they will do is, because of this enzyme, what is the function of this enzyme? This enzyme is going to break the cell wall. Okay, break the cell wall. You know that, right? In the plant, there is a cell wall is present, right? Whenever this cell wall was break, okay, cell wall was break, this tomato is going to be ripened. Okay, so that's why what we have to do, we have to remove this polygalactourase, or we have to make this polygalactourase enzyme to release slowly. Okay, not rapidly. Whenever it was rapidly increases, means rapidly the tomato is going to be ripened, right? So in the biotechnological process, what they have did is, they have made this polygalactourase enzyme to release slowly. Okay, release slowly. So then what happened? Whenever it was released slowly, so obviously the tomato is going to be stay, right, without the ripening. Okay. So this is the one of the importance of the post-harvestal loss. Okay. Students, if you are getting any confusion, please tell me. Okay. Srinivas Rao and Jainvi Middle Andaman, Jainvi Chitradurga, admin. Okay. Please tell me if you have any confusions. Okay. Okay, this is the GMO plant. Okay, GMO tomato. So this tomato is having the name, student. Okay, this tomato name, we are given that flavor savors. Okay, this tomato name is flavor savar. Okay, they will ask you. So which is the name of the flavor savar? So that is a genetically modified tomato. Okay, so the enzyme is polygalactourase or uronase. Okay. Okay, so in the biotechnological application, we have created the pest resistant plant and the insect resistant plant. Okay, so for example, you can see here we have created the BT cotton and BT corn, rice and tomato, potato, soya bean, etc. So which is having the pest resistant. What is this pest resistant, student? What is this pest resistant? I told you in the agrochemical method, right? So to infect, whenever what happens, the pest is going to infect this plant, right? So to keep our plant away from this pest, so we are going to we are going to apply in the agrochemical based method. What we are going to do, we are going to apply the pesticides, right? So, but in the agriculture, in the biotechnological process, what we are going to do is, we are genetically modifying that to carrying the pest resistant plant. Okay, so we'll study about this in the next slide. Okay, so see here. 1 minute. 1 minute, give me time. Okay, so here in the pest resistant, you can take an example of BT cotton. Okay, so can you tell me what do you mean by BT cotton? Can you tell me, students, what do you mean by BT cotton? The full form of BT is Bacillus thuringiensis. Bacillus thuringiensis. Okay, 1 minute. Bacillus thuringiensis. Thuringiensis. So student, what is this Bacillus thuringiensis? This is a bacteria. Okay. So here you can observe here, there is a bacteria, right? So here it is, you can see bacteria is there. So this bacterial name is Bacillus thuringiensis. Okay. In this bacteria, it is having some gene. Okay. This gene, the Bacillus thuringiensis, BT I will write. Okay. Bacillus thuringiensis is going to carry some gene. Okay. That gene name is crygene. Okay. So this crygene is going to produce some prototoxin. Okay. Prototoxin. Okay. So whenever this prototoxin was synthesized inside the bacteria, it is not at all harmful to that bacteria. Okay. Not at all harmful to that bacteria. So whenever any insect or any pest feed on this bacteria, okay, whenever this insect, this feed on this prototoxin, okay, you can take any insect or pest, okay, insect or pest, if it is feeds on this prototoxin is in the inactive form, right? Whenever it is in bacteria, it is in the inactive form, right? So now this prototoxin is going to be activate, okay, activate, and it is going to be converted into the toxin. So how it is going to convert? So whenever this pest was or the insect was eaten this, right? So in the gut, okay, in the gut of that pest or the insect, so there will be the alkaline pH will be there, okay, alkaline pH. What do you mean by alkaline pH? You know that, right? So above the 7 pH, we will call it as the alkaline pH, right? 7 pH, right? So in the alkaline pH, this enzyme is going to be activate. So this toxin is going to be activate. Okay. So this is the main thing. Okay. So now I will tell you how it is useful. Okay. See here, this toxin is going to be activate. Then this toxin is going to be bind on the epithelial, okay, epithelial cells in the midgut, in midguts. Okay, epithelial cells in the midgut. In which midgut it is going to be bind, student? In which midgut? In the insect or in the pest. Okay, so you can take insect. Okay, in the insect midgut, it is going to be bind on the epithelial cells. Okay, so now what happened? Simply it will bind? No. Okay, if it is simply bind means what happened? Nothing will be happen, right? But this toxin is going to be create pores on the cell. Okay, create the pores on the cell. See here. So this is a cell, right? So this is a cell. You can see here. This is an extra fluid. Okay, extracellular fluid. This. Okay. So this cell is there. So what happened if the pores was created in our cell means? What happens, student? If the pores was created in the cell means? So the extracellular fluid is there, right? The extracellular fluid in the outside is there. So this is come inside, right? So it is going to cause the osmotic imbalance. Osmotic imbalance. What do you mean by osmotic imbalance, student? What do you mean by osmotic imbalance? We know our cell is going to be maintain the osmotic balance, right? Osmotic balance. The salt concentration and the liquid concentration should be maintained. So whenever it created the pores, so the salt from the outside, this is the outside, right? So here you can see from here, the salt will go inside the cell and the liquid will go inside the cell. It is going to cause the osmotic imbalance and it is going to be burst. Okay. So what happened? So it is, if the cell, it is going to have only this much of the liquid. So now what happened? From the pore is created, all the things is comes inside the cell and it is going to be increases in the size. Finally, it is going to be burst, right? So this is how the epithelial cells in the midgut is going to be damaged. And so see here, it is going to be create pore inside the epithelial cell. Where it is going to create the pore? In the epithelial cells, right? In the epithelial cells, it is going to create a pores and osmotic imbalance is going to be happen. Osmotic imbalance. After that, what happened? Cell lysis, right? Cell lysis, or we will call it as a cell is going to be burst, right? So this is what happened in the BT cotton, or we can tell about the, so this is the importance of the prototoxin here. I think you understood this, right? I will tell you in detail, okay? Okay, this is what about the Bt toxin I am telling. Okay, what is the importance of Bt toxin? So now I will tell you how they are used in the agriculture. Okay, so I will revise one thing. See here, Bacillus thuringiensis. From the Bacillus thuringiensis bacteria, we are taking the crygene. So what is the importance of that crygene? It is going to produce some cry protein. Okay, cry protein, or we can call it as the prototoxin. Okay, so whenever it is in the bacteria, it is inactive. So whenever the pest or the insect, it is not at all going to harm to the bacteria. It is not going to be not harm to bacteria, not harm to bacteria. Okay. So whenever this pest or insect ingested on that bacteria, or it takes this prototoxin in the gut of that bacteria, in the gut of that insect or the pest, this is going to be activated. So which is the activation form? Toxin. So that is going to be, toxin is going to be activated. So then the activated toxin will go and bind to the epithelial cell. So when it binds to the epithelial cell, it is going to create a pores, and means that I have told you, it is going to create a pores, and it is going to cause the osmotic imbalance. Thereby it is going to help in the bursting of the cell. Not helping, it is going to cause the bursting of the cells, right? So thereby the insect is going to die, right? Thereby it is going to cause the death of insect, right? It is going to cause the death of insect. Okay. Okay. So then how we are going to use this? Okay, in the biotechnology. So see here. First we are going to take a crygene. Okay. So student, here you have to remember that. 1 minute. Do you understood this, student? Can you tell me whether yes or no, whether you understood this or not? Can you tell me? In the chat box, you can answer me. Okay. So yes, JNV, Middle Andaman. Okay, fine. So let us see which are the bacteria. So which are the, okay, 1 minute. Yes, thank you, Srinivas Rao. Okay, so let us talk about, okay, not cry, 1 minute. Okay, so which are the types of the insect is going to be cause harm to the Bt. Okay, that is going to cause harm to the cotton. Okay, so let us study about that. So here you have to remember this Lepidopterans. Okay, Lepidopterans. Okay, Lepidopterans is the first, and second one is the Coleopterans. Coleopterans, and third one is the Dipterans. Okay, third one is the Dipterans. Okay, so by these three groups of the insect, the cotton plant is going to be damaged, or it is going to give a low yield. Okay, see here in the Lepidopterans, it will come the budworm and the armyworm. Okay, so that is the budworm and armyworm. Armyworm. Okay, remember that. Any worm is there means, so that is the Lepidopterans. Okay, thank you, admin. Okay, so then we will come to the Coleopterans. Okay, okay, 1 minute. In the Coleopterans, we can study. So in the Coleopterans, it will be the beetles. Okay, and in the Dipterans, can you give the example, students? Flies and very good, mosquito, right? Yes, flies and mosquito will come under the Dipterans, right? So Lepidopterans, Coleopterans, and the Dipterans. Okay, so remember the flies and mosquito will come under the Dipterans. They can give you this in the multiple choice. Okay, so that's why you have to remember this. Okay, so now let us see how they have inserted this. Okay, so you know, student, first we have to produce the recombinant DNA, right? First we have to produce the recombinant DNA. How we are going to produce? So first we have to insert the genes, right? So you know how to we produce a recombinant DNA. So let us study which are the genes we will use for the which plant. Okay, so see here, this thing important, you have to remember the types of the genes. Okay, see here, there are three types of the genes. Okay, three types of the genes. Okay, so see here, which are these three types means. First one is the Cry1AC. Okay, Cry1AC and the Cry2AB. Okay, Cry1AC and the Cry2AB. Can you tell me another? Cry1AB. Okay, 1 minute. Sorry, 1 minute. Cry1AB. Okay. So student, here you have to remember these 3 genes. Okay. So 1 minute. My network is low, so that's why I'm offing video. Okay. So Cry1AB. See here, Cry1AC and the Cry1AB. So we will use it for the bollworm. Okay. So we will use it for the cotton. Okay. Cotton bollworm. Bollworm. Okay. Cotton bollworm. Okay. And Cry1AB, we will use it for the corn. Okay. For the corn borer. Okay, for the corn borer. So these 3 genes you have to remember, students. So that is the Cry1AC and Cry2AB for the cotton plant. And if we want the pest resistance in the corn means, we will use the Cry1AB gene. Okay, in the cotton means Cry1AC we can insert, or we can insert the Cry2AB. Okay, so see here how we are going to produce the recombinant DNA. You know that, right? So from the bacteria, we will take the plasmid, right? From the bacteria, we are going to take the plasmid, and from the gene of interest, from the bacteria, right? From the bacteria, Bacillus thuringiensis, we are going to take the Cry gene, right? For example, I am inserting this gene into the corn. Okay, corn. Which gene I have to take? Now I'm inserting this gene into the corn. Which gene I can take, student? Can you tell me? Which gene I can take for the corn? I need answer from you all. Which gene I can take for the corn? Now I am inserting this. Now I am making this corn to the pest resistance. Which gene I can add? Cry1AB, right? Cry1AB. See here. Cry1AB I am going to add. Cry1AB I am going to take. Okay. So now I am going to insert this Cry1AB. I will take the yellow color. Okay. So Cry1AB. So this will be the yellow color. Okay. Cry1AB. So then it has become the recombinant DNA, right? So now it is recombinant DNA, right? So this is the recombinant DNA. Recombinant DNA. So now this recombinant DNA, I am going to insert through the agrobacterium. Agrobacterium into the plant, right? To the plant, through the agrobacterium, I am going to insert this into the plant. Okay. So now see here, now our plant is going to produce a, now our plant is going to started to produce the Cry. Okay. Cry gene was inserted. Now it is going to started to produce the prototoxin. Here you have to remember that for that prototoxin, it is not going to harm our plant also, right? So it is not going to harm our plant also means it is in the prototoxin. So that is the inactive form, right? See here. So now we have inserted it. See this picture. This prototoxin was inserted into the BT corn. Okay. See here in the BT cotton also. Okay. See here now. This BT corn is there, right? So now insect was bite. Okay. Insect was bite. In the saliva, along with its saliva, it is going to take the what? It is going to take the BT toxin from the plant. Okay. It is going to take a BT toxin. So now what will happen? See, a BT toxin was inside the bacteria. Sorry, pest or the insect. Okay, you can take a pest. So now what happened? This BT toxin, see here, it is in purple color, right? It is in the inactive. Whenever it is in the purple color, it is the inactive, right? So now it is converted into the blue color. So that is the active toxin form, right? See here now, this active toxin form, here you can see this is a epithelial cell. Okay, epithelial cell. So on the epithelial cell, this toxin is going to be bind. So this you can see, this is the cilia, this is the epithelial cell on the midgut. Okay. So this toxin is going to be bind, and you can see here. So this is not important. So only this thing you can see. It is going to bind on the epithelial cell. So then it is going to cause the pores. So that do you mean by pores means it is going to cause the membrane insertion. See here you can see pores leads to the osmotic cell lysis, right? I have told you before only, right? Osmotic imbalance. So then what happened? So the cell death is going to be happen. Okay. So this is what about the pest resistance or the BT cotton. Okay. So I think if you understood this means you can give me a thumbs up. Then I can understand that whether you understand or not. Okay. Okay. So next we will go to the this BT cotton now only we have studied, right? So no problem. Then here is the question for you, student. So BT toxin is harmful to the insect-like. Okay, insect-like. So option A, lepidopterans. Option B, coleopterans. Option C, dipterans. You can answer me. Okay, you can answer me. I will run the poll. Yeah, you can answer me in the poll. I need answer from you all. No one still answered. Now only I have given that to you also, right? Answer me, student. This is the correct answer. Very good, very good. Very good. I'm getting the answer. Very good. I need an answer. I'm going to end the poll. Okay. Yeah. So I'm going to share the result with you. Yes. So you are given the answer. So that is the option D. Yes. So option D is the correct answer. Okay. So yes. So all of the above is the correct answer. Okay. 1 minute. All of the above is the correct answer. Okay. Okay, so now let us come to the pest resistant plant. So that is the nematode resistance in the tobacco plant. Okay, tobacco plant. So students, here you have to remember that. So nematode is the parasite. Okay, nematode is the parasite. You know that what do you mean by parasite, right? So it is the parasite. It will be the heterotrophic. Heterotrophic means what? It is completely depend on the other organism for the nutrition. Okay. Completely depend on other organism for nutrition. Other organism, right? For what? For nutrition. Right. Okay. So see here, this tobacco plant. So this nematode is going to affect the roots of the tobacco plant. Okay. So see here that the name of that nematode is Meloidigne incognita. Okay, Meloidigne. Sorry, 1 minute. Meloidigne incognita. Meloidigne incognita. See here. So this process. So which biological process we have used is biotechnological process is the RNA interference. Okay, RNA interference. We will also call it as the RNAi. Okay, so that is the RNA interference. So student, here you have to remember that RNA interference is going to be happen only in eukaryotes. Okay, it is not going to be happening prokaryotes. Okay, it is going to be happen only in the eukaryotes. Okay, this is the type of. It is a type of cellular defense. What is the cellular defense? Whenever some foreign particle or the foreign RNA or DNA ingested into our cell means our cell, there is some immune, our cell, some proteins are there. It is going to be digest that protein. Okay, it is going to digest that protein. So that is the thing we will do in the RNA interference. Okay, so let us study how we have used that process in the nematode resistance in the tobacco. Let us see here. Okay. So student, before going to do that, so what we will do, we are going to identify one gene. Okay. Identify the gene in the Meloidigne incognita. So without that gene or without that gene product or the protein, the Meloidigne incognita or the parasite, it is not going to be live. Okay, it is not going to be live, or it is going to cause the death of that organism. Okay. For example, if you take the plants, in the plant, the important enzyme is the rubisco, right? Without the rubisco, is that plant is going to be live? Without the rubisco, is that plant is going to be live? No, right? So rubisco is very important for the photosynthesis, right? So like that, the specific enzyme, okay, without that enzyme, this nematode will not survive. That gene we are going to be identify. For example, you can see this is a gene, okay, A gene, okay. Without this gene, the nematode is not going to be survive, okay. So this is the gene we have taken, okay. This is the gene we have taken, okay. So this we will take, we will convert it into the mRNA. So obviously we will not take the gene. We are going to isolate the mRNA, okay. mRNA from this parasite. We are going to identify the mRNA only, okay. So mRNA we are going to take, and this mRNA is single stranded, right? This is the single stranded, right? So we are converted into the double stranded. Okay, we are going to convert into the double stranded. So now our double stranded RNA is ready, right? What we are going to do now is this double stranded RNA, we are injecting that into the plant. Okay, so we are inserting into the plant. Inserting into plant. To where we are going to insert, students? Plant. So in all the things, we are going to insert the DNA, right? So, but here we are going to insert the RNA. Okay. RNA we are going to insert. So then we have to observe here. So there is a cellular defense, I have told you, right? So what is the cellular defense? If anything, if any person, unknown person come to our home, can we going to greet them? We are going to give some, we are going to give a juice to them, and we are going to tell them, sit down, and we know you, this all the thing we are going to tell? No, right? Because we don't know that person. If that person is known, we will give some juice and we'll greet him, right? If the person is not known, how we are going to talk with him? No, right? Or if any bad persons come to our home, we don't know anything about that villain or any terrorist, means what we will do, right? So the same thing, see here. So whenever this double-stranded RNA come inside the body, so this will started to break down that RNA. Okay, so this double-stranded RNA is there inside the plant, right? Inside the plant. See here, inside the plant, it is going to be happen inside plant. Okay, so what happens here? This double-stranded RNA was entered inside the plant, right? So there are some drosha, there are some enzymes. So that enzyme name, we will call it as a drosha. Okay, drosha. We are given the name for the enzyme, drosha. So what is the function of drosha? Drosha will do the breakdown of that RNA. See here, that RNA is not ours, no, that too double-stranded. So double-stranded is not, the double-stranded RNA is not ours, right? So in our body, only the single-stranded mRNA is going to be synthesized, right? So this double-stranded RNA is not ours, then why we need it? So that's why the drosha enzyme will cut the double-stranded RNA. Okay, see here, it is going to be cut that into the double-stranded RNA. So now what will happen? So this double-stranded RNA, so this another enzyme will come. So that we will call it as the RISC. RISC, RISC. Okay, RISC means, I will write here. 1 minute. Here I can write, right? RNA induced slicing. Slice means cutting. Okay. RNA induced slicing, or we can call it as a silencing complex. Okay. You can call it as a silencing complex. Okay. 1 minute. RNA induced silencing complex. Okay. So the name of RISC is RNA induced silencing complex. Okay. So now RISC, what it will do means, so this RISC will take the only one RNA. Okay. So only one RNA. See here, it is having the 5' 3', and it is having the 3', 5'. Okay. So it will take this 3 prime to 5 prime. Okay, see here, it will take the 3 prime, 5 prime strand and it is going to form one complex. Okay, see here, it is going to form one complex. Okay, it is having, here you have to remember, it is only having the 3 prime to 5 prime RNA. Okay, so another RNA is there, right? So another RNA, so that is a 5 prime to 3 prime is there, right? So it is degraded. It is degraded. In the plant cell only, it is degraded, not required. So that's why it is degraded. Now, the important thing is 3 prime to 5 prime RNA. So that is combined with the risk complex. Okay, combined with the risk complex. Now, how it is means, for our home, some unknown person was entered. What we will do? First, we will try to fight with him. So later, the police will also join hands with us, right? So this is like, so okay, the risk complex, like the police, okay, it is joined with us. Okay, so now we become the very strong, right? So see here, now what happened? So this thing happened in the plant, right? So this is the thing we have did in the plant. So now what happened? So this Melodignae incognita is very small. Okay, it is very small. So what happened? So this will ingest this. Okay, this will ingest this small RNA with the risk complex. Okay, so this small RNA with the risk complex. Okay, so now what happened? So in the Melodignae incognita, see here, this is in the Melodignae incognita. What happening in the Melodignae incognita? Okay, see here, in the Melodignae incognita, DNA is there, right? This DNA is going to produce the mRNA. mRNA. Can you tell me, guys, what is the position of this mRNA? 5 prime, 3 prime, right? 5 prime, 3 prime. mRNA was synthesized. 5 prime, 3 prime. So now what will happen? So this risk complex is having the 3 prime to 5 prime, right? 3 prime to 5 prime. So here you have to remember that. So here it is having the both sense and the antisense RNA. Sense and antisense RNA, right? Both the sense and the antisense RNA is produced, right? So see here, this incognita mRNA is there. So, and single stranded RNA with the risk complex, which is produced in the plant. So whenever this was taken, this insect, this Melodignae was taken this. So this mRNA, single stranded mRNA, they along with this risk complex will go and bind to this mRNA. The mRNA, I have told you, right? So in the body of this parasite, the mRNA is synthesized. So that is going to give a specific protein, right? So whenever the risk complex along with its the 3 prime to 5 prime RNA, which is having the complementary, right? So 5 prime, 3 prime, it is a double stranded means the 3 prime, 5 prime is complementary, right? Complementary, it must be complementary, right? Complementary. You know what is a complementary, right? So here it is having the A means in the RNA, it is going to be have the U, right? Here it is having the C means it is having the G. It is having the U means here it is having the A, right? So it is the viral RNA. So that is the 5 prime to 3 prime, viral RNA. So this is the 3 prime to 5 prime. So this is along with the risk complex, which is produced inside the plant body. Okay, so now what will happen? So it is going to form the complementary strand. Okay, complementary strand. So now what happened? It is going to produce the double-stranded RNA, right? So now double-stranded RNA is going to be formed. The risk complex is there, right? It is going to be degrade that RNA. Degrade RNA. That is double-stranded RNA. Okay. So thereby, obviously, so this enzyme is not going to be synthesized. The parasite is going to be die because of that lack of enzyme or that protein, right? So I have told you how we are going to insert that gene. Without that gene, then that parasite is not going to be live, right? So that gene we have inserted in the plant, so that the complementary strand is going to be bind on the mRNA of the parasite. Then it is going to be cause the death of that parasite. Okay. So student, it is very difficult to understand, I know. Did you understand? Can you tell me whether understood or not? If you have any questions, you can ask me. This is a new topic to you. I think it will take a time to understand. Can you tell me whether you understood or not? You have to give reply, student. Otherwise, how can I understood that whether you understood or not? Right? Okay. So I'll revise once again. So what we are going to do in the nematode resistance in the tobacco plant? We are taking the RNA. So RNA, so which RNA? mRNA. We are going to produce the double stranded RNA. So this double stranded RNA, double stranded, okay. So we are going to insert into the plant, right? Insert into the plant. So then it is going to produce the 3 prime, 5 prime along with the risk complex, right? So here, this parasite is there, right? Parasite, it is going to produce the mRNA. So now, this mRNA is having the 5 prime to 3 prime. So this risk is having the 3 prime to 5 prime. Now it is going to form the complementary strand. So then it is going to be, double strand is going to be degraded by risk complex, right? So then it is going to cause the death of Melodegnae incognita. Okay, student? I think you understood this. Otherwise, you can tell me. So, student, let us study about the application in the medicine. So, this application in the medicine, we can start in the next class. Okay. So, other than that, if you have any confusion, means you can ask me. Other than that, if you have any confusion, means you can ask me. Student, here you have to remember. So, in the Oreasa sativa, so that is Oreasa sativa. What is the Oreasa sativa, student? So, that is the rice, right? Rice. So, in the rice, we have produced the we have inserted the beta carotene gene. Okay. Beta carotene means it is a precursor. Okay. It is a precursor for what? Vitamin A. Okay. Precursor for the vitamin A. So what is the importance of the vitamin A, students? Vitamin A is very important, right? If the vitamin A is not there, means it is going to cause the night blindness, right? Night blindness. Okay. So this beta carotene gene, which is the precursor, we are going to insert. Okay. We are going to insert into the plant. Okay. So that is going to give a vitamin A after the activation. Whenever we eat that, so it is going to give a vitamin A for that person who ate that rice. So we popularly call that as a golden rice. Okay. Popularly, we will call it as a golden rice. Why we will call it as a golden rice? We know that, student, right? So whenever it is in the green color, there is a pigment present is chlorophyll, right? So it is the yellow in color. So that is the reason we will call it as a golden rice. So what is the pigment present in the yellow color? So that is the beta carotene. Okay. So here you have to remember the golden rice, which is consisting of the gene for the precursor for the vitamin A, not for the vitamin A, precursor for the vitamin A. Okay. So that the gene we have inserted into the rice. So that is the Oreasa sativa is the beta carotene. Okay. Beta carotene. So that is going to give for the patient. So there will be night blindness. Okay. So this is about the application in the agricultural, student. If you have any questions, you can ask me. Otherwise, you can give me your feedback. Give me your feedback. How was the class? Thank you so much for attending the class. So students, if you have any questions, you can ask me in the chat box. Okay. You can give me a response in the NPS poll. Valuable feedback. If you have any questions, you can ask me. So admin, I think Srinivas Rao Pentapati is there. So you can give me your feedback. Yeah. Okay. So thank you so much, student. That's it for today. So please, whenever coming for the next class, read and come. Okay. Yeah. So thank you, Srinivas sir. Thank you. Thank you, student. Let us meet in the next class. Take care. Study hard. Bye. Take care all.